for joining. Beyond that was wonderful. I have several questions, but let's discuss offline. So we have been working heavily on the immune system, and especially to understand what happens to the immune system as we age. As you can imagine, this have a lot of implications for both health span as well as the lifespan. So I'll start with a brief intro to immune system aging. So as we age, the immune system goes through significant changes. This involves both the adaptive immunity, where we lost the naive T and B cells, for example, and we accumulate dysfunctional terminally differentiated cells and see a contraction in the T and B cell repertoire diversity. But on the other hand, if you look at the innate immunity, we also see an activation. So there are chronic activation of innate immunity, upregulation of the inflammatory responses. Some people like the term of inflammaging, which is the systemic increase in the inflammation with age. And IL-6 has been one of the very frequently referred molecules in this context. So together, these changes reduces the ability of older individuals to fight against diseases and infections. COVID-19 is a good example, but it also decreases their ability to respond to vaccination. They cannot benefit from vaccines as good as they used to, and they shorten the health and lifespan of individuals. Despite this, there was one area that has not been very well explored, and this is the differences between men and women immune system aging. So we have started this project because we really wanted to understand these differences in the sexes for various reasons, starting from life expectancy. So in US, for example, women live 5 years longer than men, but in any other country, this is the case, women live longer than men, and this is a significant difference in many cases. In addition to that, we also see sex dimorphism in immune diseases. For example, men are typically more susceptible to infectious diseases. COVID-19 is also an example of this, where the death rate for men is 1.5 to 2-fold higher compared to women. On the other hand, women are more susceptible to autoimmune diseases. For example, lupus, almost 90% of lupus patients are women. And also, as we already heard a little bit from John Sang, there are sex differences in immune responses, like vaccine responses and etc. So to be able to answer this question of the sex differences in aging, we have been collaborating closely with University of Connecticut Health Center Aging Center, which is next to our institute, as well as Jacques Bancherou's lab at JGM, which is an immunology lab. So our current cohort includes 81 men and 91 women, and these individuals are clinically screened for any confounding diseases or therapies, so they do not have any of these. But we also carefully match every male and female subject in terms of age, sex, BMI, age, BMI, and other potential clinical factors. So these individuals are recruited and split into 3 groups, young, middle-aged, and older adults after discussions with the aging center. So from every individual, we collect blood. PBMCs are isolated. So you already heard about PBMCs. There are cell compositions of different immune cell types in the blood. And we have been generating ataxic and RNA-seq from these samples to study both epigenomes and transcriptomes. So the data I present here is bulk ataxic and RNA-seq, but we are moving in the direction of single cells. And we have been also generating flow cytometry data to study the cell compositions. And my lab has been building the systems immunology pipelines to analyze and integrate these data. So all of these individuals, as I already told you, screen for confounding diseases, and we were able to match male and female subjects as much as possible. So the first question that we ask from this data are, what are the age-related epigenomic changes in PBMCs? Before I show you the data, all the analysis here, almost all the analysis here are done by Eladio Marquez, who was in my lab, who is now at Sanofi. So when we did the differential accessibility analysis in our ataxic samples, separately in men and women, comparing young and older individuals, we realized that there are 4,500 differential peaks in women, and this number is almost double 10,000 in men. So these are just MA plots. Every dot here is an ataxic peak, and if the dot has a color, it's either opening or closing. So the most striking thing here is the fact that although the cohorts are comparable, there are many more epigenomic changes in males in comparison to females. So to be able to interpret these changes that we see in the bulk ataxic data, we have been taking advantage of the roadmap epigenomics projects data. Some of you might be familiar, but this is still one of the biggest reference data sets when it comes to human epigenomics. They have profiled more than 100 cells and tissues, and they identified functional states using an algorithm called chromHMM. So we obtained these chromHMM states from the blood-derived immune cells from this website. And then we uncovered the ones which are very cell type specific, including both promoters and enhancers for the cell types which are within the PBMCs. And finally, we annotate the differential peaks opening or closing with age with these cell type specific regulatory elements. So when we have applied this to our bulk ataxic data, which is, you can consider this as a way of convoluting the bulk signal. So we realized that both in men and women, the orange is peaks which are activated or opening with age. Both in men and women, innate immunity was activated with age as we have expected, but the level of activation was much higher in men, especially for CD14 monocytes. On the other hand, T cells were declining as expected. In the case of B cells, we detected a significant difference between men and women. So the declines were very specific to, or very much more significant in men, whereas in women, we see more activation of B cells with age compared to men. So obviously, which has been already covered by Beyond to a little extent, is the cell compositional changes. So the cell compositions of PBMCs change with age. We already know that. So this is flow cytometry data from our cohort, where we split individuals based on their sex as well as the age groups. So we are just looking at the main populations here: T helper cells, cytotoxic T cells, CD14 monocytes, and the B cells. So when we did look at these changes, we observed, as expected, the T cell numbers were declining, mainly stemming from naive T cell decline. We didn't see major changes in the CD14 monocytes. On the other hand, we detected that the CD19 B cell numbers are very specifically declining here in older men after 65 years old. And this decline in the B cell numbers was also observed in another cohort that we reanalyzed the data for. And the declines are not coming from one B cell subset, it is stemming from multiple B cell subsets. And this was a conserved signature across the two cohorts. So after finding these epigenomic changes that we associate with age, as I told you, we have collected data from middle-aged individuals, which gave us an opportunity to understand whether there are certain time points in our lifespans where these changes are taking place, and whether there are differences between sexes and eventually between individuals. So to capture these, we call them temporal genomic patterns. We have put together a computational pipeline, which starts with our process ataxic and RNA-seq data after all the normalization and batch adjusting. Then we use autoregressive integrated moving average ARIMA models, which are giving us the opportunity to conduct time series analysis on these data, which are very robust statistical models for trend analysis. So we fit ARIMA models and then identify ataxic peaks or genes which has a very significant chronological trend that based on these ARIMA models. Finally, we cluster these temporal genes and peaks to identify the ones which have similar trends and functionally annotate them to be able to interpret these changes. So we apply this pipeline on our data sets. This is data from ataxic, but RNA-seq is in our paper. So this gave us 13,000 differential or chronological peaks in females and males, roughly same amount of peaks, which clustered into 3 groups. And when we annotated these 3 groups, we showed that both in females and males, the declining chromatin accessibility is linked to T cells, increasing chromatin accessibility linked to monocytes and NK cells, which is confirming our differential analysis. So after looking at these heat maps long enough, we realized that maybe there are these declines or increases in the epigenomic remodeling are not continuous. So maybe there are some time points in humans' lifespan where these changes become much more significant. And this could be clinically interesting because this could be the time points where interventions might be introduced to the individuals. So to detect this, we put together a simple computational framework. So the framework is mainly we select an age, we study 5 years before and after. We change the window size, but for the sake of the explanation, let's say we study 5 years before and after, and then calculate a p-value, shift the window, calculate a p-value, and continue until we span the whole lifespan. So the idea behind this analysis is the fact that if there is a very significant difference between the 2 windows, then we will see a jump in p-values. And when we did this, we found 2 consistent breakpoints where the p-values between the window of comparisons are really peaking. And this was surprisingly consistent across the clusters, suggesting that the cell-cell interactions might be driving some of these things. But also between men and women, there were some interesting findings. So the first time point happened in around 40 years old in both men and women, and with similar p-values. But the second time point was approximately 5 years earlier in men, mid-60s versus late 60s. And of course, also the p-value for the male differences were much more significant. So can these epigenomic breakpoints really predict disease incidence? This is again referring to some of the predictor models John Sang introduced. We don't have enough data to build machine learning models yet, but I just want to show you this really catchy figure from CDC that summarizes the top 4 causes of death. And of course, this was before COVID-19, but you can see that the start of the deaths from these 4 diseases, all of which have significant immunological components, are around 40, 45. And then there is a second time point where we see an almost exponential increase in the death from these diseases. So there is, in fact, maybe some clinical value in these chronological analysis. Finally, from these data, we asked the simple question of, are there any molecular differences between men and women at different ages? So as I told you, we split our cohort into 3 age groups: young men compared to young women, middle-aged individuals compared to each other, and then older adults. So to our surprise, when we compare the PBMCs, and of course, this is focusing on autosomal chromosomes of men and women, we realized the differences between men and women increase after 65 years old. So we didn't see major differences in younger individuals, but after 65, we detected 1,000 differential peaks. We observe exactly the same thing in the RNA-seq data. The differences between sexes increase after 65 years old. And when we annotated these, we call them female bias molecules or male bias molecules after 65. So we annotated these differences using single-cell RNA-seq-driven gene sets. Then we realized that older men have more active innate immunity, whereas older women have more active adaptive immunity. And pathway enrichment analysis also gave us some interesting molecules and pathways to look at, starting from IL-7 signaling pathway, which we have been associated to aging in a previous study. But we also observed that for male bias regions, there are a lot of inflammation-related modules and pathways. And of course, from our differential analysis with age, we know in which direction these pathways are going with age. For example, T-cell signaling is declining, whereas inflammation is increasing in both sexes. So when we put all of these together, This brings us to my summary slide, as well as the sex differences that we have observed in this cohort. So we observed an accelerated aging phenotype in men, which we have associated, or which we detected to have more accelerated increase in inflammation. And we also detected a male-specific decline in B cells, potentially stemming from cell compositional changes, as well as an accelerated decline in T cells, partially, maybe not completely, stemming from cell compositional changes. And also temporal analysis gave us interesting findings. These differences, although they are mostly consistent between sexes, they start 5 years earlier in men, which is roughly the lifespan difference between men and women in the US. And finally, our study also uncovered the older, as we age, the differences between the sexes are increasing, which was kind of unexpected because of the decline in sex hormone levels and their effect on the immune system. So older women have more active adaptive immunity. Maybe this can explain why they are able to cope with infections better, whereas older men had more active innate immunity. So to be the health span and lifespan implications of this is to be seen. So more recently, obviously, we were all mesmerized by COVID-19 and whether our findings might have a meaning in this context, especially given that COVID-19 is much more dangerous for older individuals, but mainly for older men. So we wrote this review that summarizes what we have observed in our data in terms of aging signatures in men and women, and how these might tell us a little bit about individual susceptibility to have severe COVID-19 cases. So I don't have time for it, but you can read it in your spare time. So finally, I would like to also bring this to your attention. I will introduce at the end of today too, but if you like this meeting, you'll probably like this other meeting that I'm co-organizing in September. So please take a look at this website. We have a wonderful line of speakers. It was supposed to be an in-person meeting, but it's gonna be virtual this year. So let me know if you have questions about it later on. And finally, this work has been done in very close collaboration with Jacques Banchoreau's lab, who is an immunologist in our institute. And all the sample collection is done in the University of Connecticut Health Center, Aging Center, and the data is analyzed by Eladio. So if you are interested in this kind of stuff, and if you are interested in an interdisciplinary work environment, keep our institute in mind, which is located in Farmington, Connecticut, which is doing great in terms of COVID-19 these days. All right, thank you, everyone.
Thanks, Duygu. It's a new era of advertising your place with respect to the COVID rates, I think, which is weird on its own.
You gotta use it, whatever you have.
Yeah, very interesting work. So we have one question in the QA. I'll ask you that, and then I have one, and then in the meantime, if we can have one more, we can we still have time, I think. Is ancestry also considered as the question in the QA box?
Oh, that's an interesting question. So we didn't go into the genetics in the study. I mean, it's a little bit debatable because sex is also very genetically determined. But we didn't genotype the individuals. We didn't go into this. So most of these individuals are not related to each other. We sometimes get a wife and a husband pair, but we rarely get relatives. So it's not possible to answer that question from this.
Another way of putting it would be, I guess, is your male population mimicking the female population with respect to their ancestry distribution, right? This is about ethnicity.
Oh, sorry, race and ethnicity. Yes, almost everybody is Caucasian. This is community dwelling individuals in Connecticut. Unfortunately, we have not the diverse, we do not have the diversity that we wish, but we are working on recruiting from other ethnic backgrounds.
And my question would be like, in the age of personalized medicine, I think, with respect to either vaccine design or with respect to drug, targets or therapy, different therapies. When do you think either aging or biological sex could start realistically being considered as a variant that you can consider?
That's a good question. So this has to be done hand in hand with clinicians. So for example, if we give an example from the aging individuals, like the cutoff that clinicians use to give a high dose of flu vaccine to individuals is 65. So at 65 years old, it doesn't matter if you are a man or a woman, if you have a wonderful immune system, you get the high dose vaccine. But maybe this is not needed, because maybe in women, I would argue that this should start from 70. I think it's gonna take a while, but it is great to have these collaborative teams and efforts, because every time we find something from these data, our clinicians are also on board, so they see what we see in the data, and maybe in the future, these will be reflected into the clinical decisions.
One last question. How do sex hormones influence immune cell function, and does the p-value spike in women overlap with menopause? Do you think this may explain some of the differences observed? That's
a great question. So hormones have very significant effects on the immune cells. Some of them are direct effects. They can directly bind to immune cells and affect their functions. We didn't measure the hormone levels here. So in terms of, if I go back to our breakpoints, they don't really match to the menopause. So they are quite later than the menopause age. But that's going to be a very interesting future study to really carefully study that and understand whether there is a genomic signature of menopause on the immune cells and functions. All right, perfect. I'm going to stop sharing.
Thanks a lot.